Okay, hello. Welcome to the Neuropsychopharmacology Meet the Author interview. I'm Sophia Hupalo, the NPP editorial intern, and today joining us will be Carrie Martinowicz and Chloe Jordan. Carrie Martinowicz is the social media editor, and Chloe Jordan is our special project manager. And today we'll be interviewing Dr. Morgan Zipperly. She is an MD candidate in the medical scientist training program at the University of Alabama in Birmingham. She completed her PhD in August of 2020 with Jeremy Day as her mentor. And there she studied dopamine-dependent transcriptional dynamics in striatal physiology and cocaine reward. And Dr. Zipperly is currently completing her medical school clerkship and plans to finish her MD in December of this year. And so today we're chatting with Morgan about her recent neuropsychopharmacology paper, which was published in September 2020. And the paper is called Regulation of Dopamine-Dependent Transcription and Cocaine Action by GAD45B. So I'll hand it off to Chloe with her first question.
Thanks, Sophia, and thank you, Morgan, for joining us today for one of the first Meet the Author interviews. We'll link to your article in the video description and in the notes, but it would be great if you could summarize what the major findings of your paper was recently published in NPP and how you feel like these findings influence our thinking in the field.
Thanks. Well, thank you all for having me here today. I'm really excited to get back into the basic science world and talk about research. So our paper, we've looked at the role of GAD45 beta in cocaine action, specifically acute cocaine reward and reward memory, and dopamine-dependent transcriptomics. So we found that in rats, wild-type rats, just 1 hour after having like a acute cocaine reward, classic intermediate early genes, or immediate early genes, excuse me, as well as GAD45 beta were all upregulated. And so, previous studies in the field have shown that GAD45 beta seems to be required for activity-dependent demethylation, DNA demethylation. And so, knowing that, we also looked at DNMTs and TETs and found that none of those were upregulated either 1 hour after cocaine or 24 hours after cocaine. So it seems like GAD45 beta is, induced by acute cocaine reward, but maybe not through this DNA demethylation mechanism that we were thinking at first. So to dig more into this, we next did a cocaine locomotor sensitization paradigm over where the animals had 2 days of baseline, and then 1 day where they could be exposed to cocaine, a week off, and then on day 10, again, exposed to cocaine, and found that in all of our groups that had any kind of cocaine experience, GAD45 beta mRNA was upregulated. So this was really interesting to us, especially because in our group of animals who received saline injections on the last day, if they had, a history of cocaine use and like we're just re-exposed to this environment, we also saw GAD45 beta increased again. So it seems to not only be important for this acute cocaine reward, but also involved in kind of this reward memory independent of the like direct psychoactive effects of cocaine. So next, I love behavior. So we also did a cocaine condition place preference. First using GAD45 beta knockout mice and found that this decreased cocaine place preference. And if we did the same kind of paradigm in rats, where we specifically knocked out GAD45 beta in the nucleus accumbens using CRISPR-Cas9 gene editing, we completely blocked cocaine preference, like at both 10 and 20 mg per gig. So that was pretty amazing to us to show like you really don't get any reward at all through at least the CPP paradigm when GAD45 beta is knocked out. So next we just did some in vitro studies, kind of further elucidating kind of the signaling pathways that GAD45 beta seems to be involved in. It seems to require DRD1, or the dopamine receptor type 1 activation, as well as MEK and Krebs signaling. And found out that in in vitro in our cultures, our striatal neuron cultures, where GAD45 beta was knocked down, there's pretty significant transcriptional dysregulation. So we had over 7,000 differentially expressed genes. And after KEGG analysis found that a lot of these are those that are involved in dopamine signaling and reward and addiction pathways. So we were excited to see that. Everything lines up. It's always exciting. We did look also in vitro at some electrophysiological responses to cocaine and in neurons, striatal neurons that had GAD45 beta knockdown. And we were actually pretty surprised there were very few changes. The only change that we saw was a decreased burst duration at baseline. So that was pretty interesting. We kind of expected with all these, behavioral changes and this really, significant transcriptional dysregulation, we thought we would see more at the electrophysiological level. So I do think that's something we want to dig into a little bit more. And then the last thing we did, just kind of getting back to that DNA methylation, process or mechanism that others have hypothesized and kind of dug into, we did reduced representation by sulfite sequencing and found that in striatal neurons where GAD45 beta was knocked out, the DNA methylation landscape was preserved at baseline, but dopamine-induced changes in these neurons at the CPG sites was like completely absent in the cultures that lack GAD45 beta in a pretty astounding manner. So there's a lot that we want to dig more into moving forward. But I think it's really, great that we could, we had the opportunity to build upon, some really outstanding work that had already been done in the field as far as just activity dependent changes in behavior and kind of stress responses, and kind of apply that to reward and addiction.
Okay, thanks, Morgan, for telling us a little bit about the paper and your research. I think we wanted to ask you a couple questions also about you and your career. So one thing that we were curious about and wanted to hear from you is being that with your being an MD PhD student and with aspirations to become a physician scientist, we were wondering how that specifically and your interest in becoming a physician scientist impacted or influenced your motivation for going into like what kind of basic research you were interested in, how you picked your PhD or things like that.
Yeah, so it's kind of funny. It was, I guess, a little backwards for me because I actually first started working in a neuroscience lab in college just because I knew I wanted to go to med school. And at the time I was like, I'm going to be a dermatologist, which I think everyone says like in the beginning of college who wants to go to med school. It's like the classic answer. And I was like, oh, I'll work in a lab. That'll look good on my CV. And then just really fell in love with it and like really loved, working in a lab and like, just kind of having hypotheses and kind of, going in like the next day and seeing what happened. And I also just really liked working with the rats. I thought they were very cute. And I don't know, I just enjoyed the different behavioral paradigms that we could do. And so I kind of had an existential crisis of like, should I go to grad school or med school? Didn't know MD PhD was a thing. Someone told me and I was like, what crazy person would do that? And then it turns out I'm that crazy person. So I feel like I had kind of picked the, addiction research in particular as something I wanted to do for my career, and then kind of had to figure out how to fit all that together. But now I think moving forward, it's really interesting to me because I think, addiction is incredibly widespread globally. And those numbers are only increasing, especially now with the pandemic and people being isolated. Unfortunately, we've seen increases in overdose deaths and hospitalizations due to drug use. And just from the clinical perspective, chronic drug use impacts, every organ in the body. So there's a lot of medicine that, to be done there that's really interesting. And I think also just the fact that we don't, despite the fact that this is so widespread, we don't have a lot of treatments for this disease. So as from the physician scientist standpoint, there's a lot of room for improvement there, I think, to kind of investigate new novel treatments and better serve this population.
Okay, and then just quickly following that up, I guess since now that you've returned to the clinic and are finishing your MD, what do you miss most about being in the lab?
I mean, the short answer is everything. I really miss the lab a lot. I miss, all my lab mates, and, working with Jeremy and working as a team. It's very weird, getting to that place where you defend your dissertation and you finally kind of accept like, oh, I know some things, because I feel like, a ton of imposter syndrome until then, and just getting really excited and having so many ideas, especially after writing, my thesis. I was like, I have all these ideas, all these things I want to do. And then you go back to med school where, you forgot everything from the last, 4 years ago. And so I just really miss the lab, like, having kind of ownership of a idea and, something to address as opposed to, I mean, I love the clinic too, don't get me wrong, but there's a lot to miss. And I'm excited to get back in the future.
That leads us to our last question. I know you're in the midst of your clerkships right now in medical school, but what are your future career plans once you finish your MD?
So very passionate about addiction research in medicine. So I know that will definitely be in my career moving forward. I'm trying to keep somewhat of an open mind since I haven't finished my required clerkships. But currently my plan is to match into a psychiatry residency. And there's a newer fellowship for interventional psychiatry. So I'd really, like to pursue that as well as specializing in addiction medicine. And from a research standpoint, I really would love to dig more into developing better animal models for polysubstance abuse, because I think clinically this is a huge problem, and to really like make translational therapies and have translational findings, we need models that better model human behavior, and so many humans use multiple substances as opposed to just one. And so I'd really love to dig into that and kind of look at the mechanisms underlying, are there interactions there? Are there different genes expressed? Are there different circuits activated? And I think that could really better direct making novel treatments. I'm also really interested in neuromodulation, so things like deep brain stimulation or transcranial magnetic stimulation, and so kind of optimizing those types of protocols moving forward to help treat that patient population would be a good goal of mine.
Well, that's certainly a lot of goals, and I'm sure you'll achieve many of them, if not all of them. But yeah, Morgan, thank you so much for chatting with us today. And if anyone has any follow-up questions, please feel free to reach out and respond to the post on Twitter, as well as YouTube. And thank you all again.
Yeah, thank you.
Thank you, Morgan.